Hey everybody, Professor Bob Long here doing your anatomy and physiology lectures. If you see me wearing the same clothes, I'm doing multiple videos in one day. I got a short window to try to get some of this stuff done. And I'm not Britney Spears or Cher. I don't have multiple wardrobe changes. So nonetheless, I'm going to be doing another muscle lecture. This is muscle lecture, I believe it's muscle lecture 5 or 6. Anyway, the number will be on the video on my YouTube page and on Canvas. Nonetheless, this muscle lecture is going to be over what we call myosin cycling, or the crossbridge cycling of the myosin molecule during muscle contraction. It's going to be a rather detailed lecture. So I'm going to set us up where we left off last time. Essentially what we're going to see here is we're going to see a piece of the sarcoplasmic reticulum. I'm just going to label it SR for sarcoplasmic reticulum, which we know the sarcoplasmic reticulum is loaded with calcium ions. It is a calcium reservoir and stores tons of calcium ions. Now, there are calcium ion channels here that are waiting to release the calcium, and there's calcium pumps that are going to pump the calcium back in. That is sitting over our actin molecule. Now, I'm not going to draw this out every single time because we're going to draw a series of steps across the board here. But essentially, what we're going to see is when the muscle is at rest, meaning we're not contracted, we are relaxed, I'm going to have my little actin filaments, or my G-actin molecules, with their subunits, their active sites sitting here like this. I think I drew one too many, but who cares? Now, we have the protein tropomyosin sitting over the active site. We have troponin sitting here waiting for calcium to bind. And we have our myosin heads. I'm only going to draw one myosin molecule, but I'll have my myosin heads sitting here like this. These are the myosin cross bridges. Now, before I get too far, there's a little bit of setup. The myosin molecule is actually an enzyme that can hydrolyze or break down ATP molecules. So sometimes we call that an ATPase, an enzyme that can break down ATP. As you guys know, I'm going to draw a quick review here of ATP, but ATP is essentially a double-ringed molecule of adenosine that's going to have 3 little phosphate groups attached to it. It is in these chemical bonds that we store energy. As I covered earlier in the semester, when we form a bond, it takes energy to make a bond. When we break the bond, it can release that energy. Some bonds release more energy than they make, and then it takes to make them, and so on and so forth. As you guys are aware, if I break this last phosphate molecule off, when I digest it, again, this is ATP, adenosine triphosphate, with 3 phosphates linearly here. I can break that down into a molecule of adenosine, and the adenosine would only have 2 phosphates, and it would release an inorganic phosphate, one that's not attached to an organic molecule. When I break down ATP into ADP plus the phosphate, some of the energy in that bond is released into the atmosphere or into the universe, the surrounding area, in some form. So breaking down ATP into ADP releases energy. Anytime I see ADP bound to something, I know the energy went somewhere. Knowing that, as a quick review, excuse me, when I look at my molecule of myosin, the myosin heads, they have ADP plus phosphate bound to them. And the energy that went into that was in that molecule is actually transferred to the hinge. I always think of the hinge like the string on a bow and arrow. Actually, I like the bow on a bow and arrow, or like the hammer on a pistol. If I use the energy in my muscles to pull on a bowstring, I'm loading that energy into the bow. It's called potential energy. It has the potential to do work. If I release the string, the energy in the bow is released and shoots the arrow. Or the same thing when you pull the hammer back on a pistol, you've taken some energy and put it into that little spring, and it's sitting there waiting to fire. You just have to release it by pressing the trigger. Excuse me. So now, the myosin head has a high affinity for ADP. It has a higher affinity for the active site on actin. Given the opportunity, it would release the ADP and bind to actin. If actin's not available, it just holds on to the ADP. it has an even higher affinity for the molecule ATP. So if I give it three choices, ATP, actin, the active site, or ADP, it will choose ATP first, then the active site on actin, and ADP last. With nothing else available, this is what it binds. Now, as you guys know from the previous video, all I need is an action potential, which I always abbreviate as AP, to come down and shock these calcium channels into opening. When calcium channels open, the calcium ions would leak out, bind to troponin, and cause it to change shape. Prior to that happening, we have this setup. The myosin head is sitting here holding on to ADP, troponin and tropomyosin are covering the active site on actin. So the first step on all of this is going to be called active site exposure. And this is outlined in the book and in my note set. In active site exposure, in order to expose the active site, all I need is the calcium to bind to troponin. Troponin will change shape and pull tropomyosin off the active site. I'm not going to draw both sides of this molecule in the entire setup every time so that it's a little easier for you to follow if you're drawing this. So here's one row of my actin subunits. But if you notice now that calcium is bound, the tropomyosin has been pulled off to the side because troponin has calcium bound to it. Now that the active site is exposed, my myosin head, which would be sitting here, or the cross bridges, binding to ADP and phosphate, will now release some of the energy here and cause the hinge to reach up and attach to the active site on actin, which is the next step in the process. It's called cross-bridge attachment. And all that's going to happen in this situation, I'm not even going to draw the troponin and the tropomyosin. I'm just going to draw my little actin subunits here with their active sites exposed. Essentially what's going to happen is the hinge is going to allow the myosin heads to reach up and bind. And when they do, they release the ADP plus the phosphate. And we can put a little I on there for inorganic phosphate. But now, just like I talked about in the last video, if I asked you to hold a couple of footballs under your arms and I threw you a live baby, since you have a higher affinity to save a person, you would release the footballs and grab the baby, and then you would change your conformation. Well, as soon as the myosin head attaches to the active site on actin, it releases the ADP, and it's going to do what's called a pivot, which is going to be the third step in this process, which is going to be called pivoting or myosin pivoting. And all that you're going to see is this exact same picture that I already have. Here's the same 7 little actin subunits. I think I drew 8. 1, 2, 3, 4, 5, 6. No, I got 7. But if you notice, the myosin hinge has allowed to pivot or pull this way. And since the Z line would be at the end of this actin filament, it's going to pull the Z line towards the M line, which would be over here. Essentially, we're looking at this happening. And we're seeing myosin heads attach while others are reaching out, and as soon as they release their ADP, they're going to pull the Z lines towards the M line. And they would do the same thing on the opposite side here, pulling the actin filaments, pulling the Z lines towards the M line, contracting the sarcomere. Now, because every sarcomere on the myofibril will have calcium available to it due to the structure of the muscle, then every sarcomere will contract throughout the entire length of the myofibril contracts, the muscle cell contracts, and so on and so forth. Now, once we pivot, we have a step that's going to be cross-bridge detachment. In order for the cross-bridge to detach, because the myosin heads or cross-bridges have a much higher affinity for ATP than they do the actin, if a molecule of ATP floats by, they'll let go of the actin and grab the ATP and detach. And that's exactly what you're going to see in this step, is that they will actually bind a molecule of ATP and release from the actin subunits. And then as they break down the ATP, they're going to reactivate. And essentially what we're going to see in the next step, and the last step is going to be reactivation of the myosin molecule. We're going to see it go back to the shape where it has ADP plus the phosphate bound. And here's my actin subunits. Now, if the action potential is still causing the release of calcium, when I reactivate, if I'm here, if the active sites are exposed, it will cycle again. The myosin heads will reach up and attach. Then the molecule will release the ADP and pivot. It will bind ATP and detach. It'll break down the ATP and reactivate. And it'll continue cycling like this. One myosin head cycling over and over and over as long as the active sites are exposed. And I can have several myosin heads cycling this way, burning tons of molecules of ATP. If you look at the sarcomeres on the myofibril models in lab, and you try to count the number of sarcomeres on one myofibril, plus the number of myofibrils in a muscle cell, think about the length of the size of one of those muscle cells, how many millions of molecules of ATP we're burning every fraction of a second. You can understand why we get tired when we have lots of muscle contractions. We call this the myosin cycling, or the Crossbridge cycling, for muscle contraction, and it explains where all the energy goes. Again, if we're sitting here at rest, the myosin head's holding ADP, the active sites are covered. As soon as an action potential hits the muscle cell, travels down the sarcolemma, down the T-tubules, and shocks the sarcoplasmic reticulum into releasing calcium, calcium binds to troponin, troponin changes shape and pulls tropomyosin off the active site. Once the active site is exposed, the cross bridge will attach, release ADP and pivot, bind ATP and detach, break down ATP into ADP and reset. And it'll go back to either step 1 if the active sites are still exposed, or if we've ended the release of calcium, it will just sit here waiting for the next opportunity for you to contract a muscle. That's how we burn energy in muscle contraction. It also leads us somewhat to understand the process of what's called rigor mortis. Rigor means difficult or tough or stiff. And mortis comes from the word muerte or morte, which means death. So it means dead stiff. Whenever you see an animal that has stiffened up and it's dead and the legs are sticking in the air, or a human being is all stiffened. When someone first dies, as our cells start to use up all the ATP and lack of oxygen and glucose stops the mitochondria and muscle cells from producing ATP, then the availability of ATP goes down. With no ATP available, I can't have the pumps functioning, so some of the calcium could slowly leak out, expose the active sites, the myosin heads would bind, release the ADP and pivot, but they cannot detach. So the muscles don't curl up and you don't cramp up, they just tighten in whatever position you're in, and we cannot release. That takes a period of time for the onset of rigor mortis. Over time, enzymes from vesicles inside the cell, lysozymes, will break down all the proteins, and if I chopped up all the proteins, eventually the person would go limp again, or the animal. So that explains what's going on with rigor mortis, which is some further support for the concept or the theory behind this cross-bridge cycling. Anyway, as usual, I hope you learned something. I hope you had as much fun as I did. For those of you that are in my class, this is in the note set. This is following along on page, I believe it's page 56. Page 55 was the last lecture where we went through the muscle contraction. It's all spelled out for you. Please look up the worksheets that will be provided. If they're not up yet, be patient. I'm working on all of this. Fill in the worksheet, understand the concept, and there'll be some quizzes coming up online in the near future. Again, this is all due to this coronavirus outbreak, and we're having to do everything online. So I'm doing a series of videos this way. I hope this was helpful. Those of you that are not in my class, maybe you learned something. Maybe it was a little easier to understand, or maybe my lectures were terrible. I don't know. Whatever you think. All right? See ya.